A little bit introduction of this topic. The fermentation-based synthesis of biocolorants provides an environmentally friendly and sustainable alternative to synthetic dyes, which are frequently connected with environmental and health risks. Natural colors can be efficiently created under regulated conditions with microorganisms such as fungi, bacteria, and yeast. This study focuses on optimizing fermentation settings to increase biocolorants yield, emphasizing its potential application in the food, textile, and pharmaceutical industries.
So in this slide, I'm going to present what is the fermentation technology used. Fermentation technology is defined as a field that involves the use of microbial enzymes for production of compounds that have application within the energy production, material, pharmaceutical industry, chemical, and food industry. Fermentation technology involved within a microorganism such as bacteria, fungi, or yeast to convert organic substrate into valuable product under controlled conditions. The fermentation technology used for the synthesis of biocolorant depends on the microorganism, the pigment type, and the production scale. Below are the primary technologies applied, which is submerged fermentation, solid-state fermentation, fed-bed fermentation, two-state fermentation for microalgae, and mixed culture fermentation. Industrial fermentation technology play a pivotal role in the synthesis of biocolorants such as carotenoids and astaxanthin, employing microorganisms like bacteria, fungi, yeast, and microalgae. The synthesis of biocolorant refers to the production of natural pigment derived from biology sources such as microorganisms, plants, and algae. These pigments such as carotenoid, anthocyanin, astaxanthin, and others are widely used in food, cosmetic, pharmaceutical, and textile as an alternative to synthetic dyes. Owing to their biodegradability, non-toxicity, and environmental friendliness, the microorganisms are cultivated under controlled conditions to produce pigment. So next, I'm going to talk about the discovery and production process. Biocolorants are natural pigments derived from biological sources such as microorganisms, plants, and algae. They include carotenoids and astaxanthins, which are widely used in food, cosmetic, and pharmaceutical due to their antioxidant properties and eco-friendliness. Microbial and enzymatic production involve leveraging the metabolic capability of microorganisms or their enzyme to synthesize these pigments under controlled fermentation conditions. For microbial production, microbial fermentation harnesses the metabolic pathway of selected organisms capable of producing carotenoid and astaxanthins. So the microorganisms include yeast, fungi, bacteria, and microalgae. So for biosynthesis pathway, carotenoid and astaxanthins are synthesized through the isoprenoid pathway in microorganisms. First is initiation, which is started with the precursor molecule acetyl coenzyme or pyruvate, which enter the methylmalonate MVA or methyl erythryl phosphate MEP pathway. Second step is formation of IPP and DMAPP. These are the building block of isoprenoid. Both IPP and DMAPP serve as the basic building block for isoprenoid biosynthesis. And the third step is geranyl pyrophosphate GPP. IPP and DMAPP are condensed to form GPP, which is the precursor of carotenoid. And the fourth is carotenoid synthesis. GGPP is cycled to produce carotenoid like beta-carotene, lycopene, and lutein. And last is the astaxanthin production. Enzymatic hydrolysis and oxidation step convert beta-carotene to extant. So that is the biosynthesis pathway for carotene and astaxanthins.
There are few ways to optimize the production of biocolorants. One of it is the selection of microorganisms. Select strains with a high pigment production potential, such as Monascus purpurus for red pigments, Spirulina for phycocyanid, or Penicillium for yellow pigments. Next is the fermentation parameters, that is divided into 5. The first one is the substrate. To improve pigment synthesis, optimize the carbon, such as glucose or glycerol, and nitrogen sources, such as peptone and ammonium salts. For the pH, adjust pH values to meet the microorganisms' development requirements and increase pigment formation. The third one is the temperature. Determine the optimal fermentation temperature for maximal production. For example, 25 to 30 degrees Celsius for several fungi. For the aeration and agitation, regulate oxygen levels and mixing to optimize nutrition intake and pigment production. Lastly is the incubation time. Determine the best fermentation length to avoid deterioration and lower productivity. For process design, compare batch, fed batch, and continuous fermentation methods to determine the most efficient system. Scale from lab to pilot or industrial scale with minimal loss of yield and quality. Then, enhancing yield. Utilize metabolic engineering or mutation approaches to enhance pigment production pathways. Supplement cultures with precursors or additions that stimulate pigment formation. For example, specific amino acids or trace metals. In terms of purification and stability, create effective downstream processes for pigment extraction and purification. Then improve stability by encapsulating pigments or adjusting storage conditions. Last but not least, the sustainability. Use agro-industrial waste or waste materials as substrates to save money and reduce environmental effect.
Biomass removal is an essential step in the downstream processing of biocolorants. It focuses on separating solid materials such as plant residues or microbial cells from the liquid containing the pigments. Filtration is commonly used to remove the solid biomass by passing the mixture through a filter, and it will leave behind a pigment-rich liquid. For mixtures where solids are difficult to settle or filter, centrifugation is employed using high-speed spinning to separate the solid and liquid components. In cases where pigments are trapped inside the cells, cell disruption methods are applied. The bead mill techniques will break the cells by shaking them with quartz or metal beads in a closed container. Alternatively, chemical lysis will use enzymes like cellulase to weaken and break down cell walls, and it will release the pigments. These processes will ensure effective biomass removal and enable further purification and processing of the biocolorants. The extraction process for biocolorants involves isolating pigments from their natural resources such as plants, algae, or microorganisms using suitable methods and solvent. In conventional solvent-based extraction, polar solvents like water and ethanol are used for water-soluble pigments such as ethocyanins and betalains, while non-polar solvents like hexane and acetone target fat-soluble pigments such as carotenoids. This method is straightforward and effective, but may pose environmental concern and risk of heat damage to sensitive pigments. To address these challenges, advanced green methods are often employed. For example, ultrasound-assisted extraction (UAE) utilizes ultrasonic wave to break open cells, enabling faster solvent penetration and improved pigment yield. Similarly, pressurized liquid extraction (PLE) operates at elevated pressure and temperature, reducing the need for large quantities of solvent while enhancing the efficiency. The choice of method depends on the type of pigment, source material, and environmental consideration. Stabilizers such as antioxidants are often added during the process to prevent pigment degradation. Once extracted, the liquid containing the pigments undergoes further stages including filtration and purification to prepare for drying and storage. These steps ensure the pigments are stable, vibrant, and suitable for application in industries like food, cosmetics, and textiles. Drying and storage are crucial steps in preserving the quality and stability of biocolorants after extraction. Drying involves removing moisture to convert the pigments into a stable and usable form. Common methods include hot air drying, which is suitable for bulk drying but it may degrade the heat-sensitive pigments. Vacuum drying is a gentler method that reduces the boiling point of water and it will minimize the pigment damage. For the best preservation of quality, freeze drying is often used as it simply melts water under low pressure and temperature. It will maintain the pigment's color and bioactivity. Once dried, proper storage is essential to ensure the pigments remain stable over time. Controlled atmosphere using inert gases like nitrogen will help to minimize the oxidation, while storage in cool, dark conditions will enhance the longevity. Additionally, packaging materials that are UV resistant and moisture-proof are often employed to protect pigments from environmental factors. These steps ensure the biocolorants retain their functionality and are ready for the application in various industries.
One of the side products of biocolorant is microbial fermentation always produces organic acids such as lactic acid and acetic acid. Next, ethanol and other alcohol was produced during fermentation as it was an anaerobic respiration of microorganism product. Then, after pigment extraction process, there will always significant side product of leftover microbial, which called residual biomass. After that, the application of biocolorant. First, it can act as food preservative. It is to protect the microbial spoilage. For example, carotenoids can act as sunscreen to maintain the quality of food from sun. Next, it can act as quality control markers. For example, anthocyanin is used as indicator to determine food quality. Last but not least, nutritional supplements. For example, riboflavin is natural food-grade biocolorant that is essential vitamin source. There are several impacts of biocolorant to environment. First, it can reduce water pollution as the biocolorant is biodegradable and cause less harm to environment than synthetic dyes, which has more toxic compound. Next, it can reduce greenhouse gas emission as the biocolorant produce minimal carbon gas to environment. Next, it is only need low energy consumption, especially energy required to recycle resource, which can result reduction of environment impact. After that, byproduct can be recovered in different industry, such as lactic acid act as precursor for biodegradable plastic and ethanol act as biofuel. Next, adjusting the parameters in fermentation such as pH and temperature to reduce the unwanted side product.
At last, the synthesis of biocolorants through fermentation demonstrates a sustainable and environmentally friendly alternative to synthetic dyes. Optimizing fermentation conditions such as pH, temperature, substrate concentration, and microorganism selection can result in higher yields and more vibrant colorant properties. This process not only reduces environmental pollution but also utilizes renewable resources, making it economically viable and environmentally beneficial. Furthermore, biocolorants provide significant advantages in biocompatibility and safety, particularly in applications like food, cosmetics, and textiles, aligning with global sustainable goals.